Hello, my name is Engr, and today I'm going to talk about Dr. Guevara's presentation at the courtesy of the Bruker Company, Novel Concepts in Mechanobiology from Individual Cells to Cancer Tissues. At first, I'm going to introduce Dr. Guevara. After that, I'm going to define AFM and the cytoskeleton, then the cell mechanics, and what the data analysis of AFM can look like. Dr. Guevara has a bachelor's degree in physics and a PhD in cellular biophysics. She's the leader of the Mechanobiology laboratory in Barcelona, where she's also teaching. The atomic force microscopies, which I will from now on call AFM, are studies on cells, especially on the cytoskeleton. Other researchers from her and her team is developing image analysis tools, then also image-based diagnostics and drug screenings with the cytoskeleton, and they are researching the cytoskeleton as biomarker for health and disease. So what is she doing when she does AFM? Mostly, she measures the mechanical properties of living cells by indenting them. She uses the AFM as the indenter and at first goes up and down the sample to get the retraction curve here. After finding the contact point, they start indenting and getting the stiffness curve. Samples with higher stiffness have larger slopes, and samples with softer stiffness have smaller slopes. When working on cells, have in mind that they are working on nanoscales and that the tips are not very sharp. Mostly they are blunted. The stiffness of the cantilever is also soft compared to the cell. So in general, we can say that she studies the mechanical properties of the cytoskeleton. So why the cytoskeleton? It provides shape and mechanical stability to the cells and is involved in the cell's functions, fate, and behavior, like migration, division, and differentiation. It's also involved in physio and pathological states, so also the development, the body movement, wound healing, cancer, and inflammation. It can also be used as a biomarker for disease diagnostics and drug discovery. There are three cornerstones in cell biomechanics. The first one is having a good bio-AFM setup, which consists of stability and minimal noise, temperature control for living cells, large piezo range, and optical integration. The second one is a suitable contact mechanics model, so Hertz-like models and beyond infinite semispace. It's homogeneous, pure elastic, isotropic, and linear elastic. The third part, which her and her team concentrates on, is the optimal data analysis pipeline. So on automation, determination of defection sensitivity, determination of contact point, and correction for tilt and signal. And that's also the point on which we are concentrating today. So why do we use data analysis? First, to get better data and therefore improve calibrations and reduce artifacts. The second reason is to obtain more mechanical parameters to further our biological understanding. And the third one is to tap into emerging fields such as machine learning. So to measure mechanics, we need to link the force which is applied on the sample to the deformation which the sample gets from the force. The AFM doesn't directly measure force and this doesn't directly measure indentation of the sample. So we shouldn't be confused about deformation or deflection of the cantilever. So what does AFM truly measure? It measures the voltage difference in the quadrant detector. There are two approaches to measure deflection sensitivity, which are both dependent on the calibration step. So the first approach contains 3 steps. Find an empty region and obtain some FD courses. Use the linear fit to compute the slope, and then inverse the slope to find the deflective sensitivity. The second approach would be taking a fit on force feedback mode, including an empty region. Then split the data into points between cell and background. Use the linear fit to compute the slope and contact point of background, and then also inverse the slope to find the deflective sensitivity. So here you can see how morning could look like when she has to measure the deflection sensitivity, and a drift in a series with the same sample is comparable to changes in deflection sensitivity when exchanging samples. There are also different approaches to find the contact point, and when you try to find the contact point, you will get a curve. You can find more than one peak, and depending on how you find the contact point, the Young's modulus will change. So to get more data, there are other 2 approaches which she calls depth sensing. She divides it into sections based on force, which you can see on the left side, where she cuts the data depending on the force between low loads and high loads. And the second approach would be cutting the data based on indentation. And so both approaches are good depending on what the focus of the research is. So often they are dealing with a 2D grid of 1D signals. And what they will get is several force displacement curves from where they will get the contact points in Z direction. After that, you can get the image of the Z extension at the contact point, and filter the image, smooth this, and refine the contact points. So what you can get after the refining is a multiplex of, for example, the height, the Young's modulus, the viscoelasticity, and the adhesion. So you will get more data through better analysis. So the first would be through depth sensing. You can get Young's modulus of causal actin and Young's modulus of deeper structures like stress fibers and vimentin. Another data you can get is the viscosity with the classic protocol of force displacement curves. And the third one would be through topography or image style analysis. You can get the Young's modulus of the nucleus and also the Young's modulus of the cell body or the lamellipodia. What can you get out of all of this? So you can get a multiplex for also machine learning classifications, which he uses for her research against for the human fibroblasts from breast cancer. And the useful classifications parameters for the classifications is, for example, the mechanics of actin cortex and its linkage to the membrane, or like also the overall cell mechanics and also the mechanics of the nucleus. So here you can see my reference slide. Thank you for watching the video. and also thanks to Dr. Gavarian, who made her presentation available for us.